Thank you, Elias, for the invitation. It's very nice to be with you again. Sorry, my Spanish is not good enough, so I have to do it in English, but I can understand well everything. And it's also a real pleasure to honor Dr. Alberto Casarin, who I had the pleasure to meet during my visit in Mexico during the first
Yeah.
NAEQUINA. So thank you again for this invitation.
Thank you, Dr. Marcio. It's yours.
So let me just try to share. Please let me know if you can see my presentation and if everything is working well.
It's working well, doctor.
Good. So without further ado, we can start. Um, Before starting, I would like to disclose that I'm going to be presenting you some unpublished data, so please don't share with anybody else, and also be mindful when listening to that because this data has not been yet reviewed by the smart people that are the reviewers of journals. So please caution when interpreting my my talk. And I was asked to share some of my knowledge and my research that I've been doing, more focused on the intestinal markers to detect intestinal disease or injuries. And I decided to divide this presentation into the traditional markers that you are familiar with, and then talk to you a little bit about the searching of new markers, and then we're going to discuss what are the future perspectives for those markers. So to start, I think everybody's familiar with the antemortem test that we do in clinics. The most used, I consider to be the blood lactate, which is a proxy for tissue injury or hypoxia. And the problem with the L-lactate is that it's not specific for intestinal injuries. It's widely used. It's a good estimator of severity and sometimes prognosis, but we cannot really tell that this is coming from the intestine. One other possibility is to visualize the gut by use the ultrasound. The problem with that test is that normally the lesions become visible only at the advanced stages of the diseases. For example, in this case of a horse with IBD, which is inflammatory bowel disease, basically infiltration of lymph of white blood cells, different kinds, into the intestine, like it was nicely explained by Dr. Danesi yesterday. So at this stage, you can actually see the lesions, but not at the initial stages of the diseases. Then we have the endoscopy, where you can go with a camera and look into the intestine, but this is limited to the proximal compartments like the stomach and duodenum. Then, in very rare situations, we can justify doing surgery to get a biopsy, which can be very informative, but at the same time, it's very invasive and expensive procedure. Now, I also wanted to share with you this cool paper that has been published where they used a capsule endoscopy. It's basically a camera, a micro camera that you can give to the horse, and there you can visualize the compartments. But again, this is a little bit expensive, and they had problems into retrieving the capsules in feces. And more or less on the same design, we are doing a study now using capsules that are intended for human use, where those capsules, after digestion of the external membrane, they can capture the intestinal content and stabilize the microbiome of that environment. And then later we can do microbiota analysis, which will be in align with what we're going to be talking later. But sometimes we don't realize how much feces one horse can do. And we read in the books that it takes 48 hours for them to pass the ingesta. that they just ate, and it became a big challenge to find those capsules into the manure. So we had troubles into finding all the capsules that we administered, and we ended up trying to develop a new method using X-rays, just like the other group from Calgary using the cameras did. And it's very hard to see, but after that we had some success in retrieving some, but not all of the capsules. So the samples are submitted for microbiota analysis, and I hope that we can share some more results of this study soon. Now, when we talk about markers of intestinal integrity, what we want is really to assess how well this intestine is absorbing things. And if we find anything that belongs to the intestine in blood, that's not a good sign. And if you find anything that belongs in blood into the intestine, that's neither a good sign. So for example, we have been using the glucose absorption test where you give some sugar intragastrically to horses and then see how the sugar, the glucose increases in blood. That's a good measure to see if the permeability of the gut or if the absorption of these molecules are good. And this has been used and validated in horses. What we have been using in other species like dogs and humans is serum ihexol, which is an iodine compound that should not be absorbed by the gut. So if you give that orally and you measure in the serum and it's present, that's a sign that the integrity of the intestinal barrier has been compromised. It has been validated to horses, so we have some reference intervals, but it's not something that I see people using in the clinics. The same thing for calprotectin, which is a protein produced mainly by neutrophils that should not be in the gut. So if you find that in feces, this could be a proxy of intestinal inflammation. This has been also validated by the group of Annette McCoy from Illinois, and we also have some references, some reference intervals if you are interested in starting using that in clinics. And then finally, we have fecal occult blood, which depending to who you ask, they're gonna say has a good or a low sensitivity, but it's a little bit controversial still to use that because, as we know, many of horses with severe colitis, for example, they still can test negative for this test. Now we have some new markers that people are trying to develop. This is out from a group in France who I was happy and lucky to participate in this PhD defense. And what they did was to induce dysbiosis by antibiotics or by grain overload. And what they found is that some fatty acids, they were increased in blood. And those are from bacterial origin, and meaning that the intestinal barrier could be compromised, and they hypothesized that it could be because also of intestinal dysbiosis. And the problem is that this is still at the beginning. This was one-time study. There are no reference values for those bioacids, and also it's very, very expensive, so it cannot be justified to be used clinically right now. And based on the same principle, we also, in collaboration with a group in Brazil from USP, what we did, I'm not going to share too much details of the study, but what we did was to try to measure the total bacteria in blood after we induced dysbiosis by starch overload. Here on day 0, we induced the dysbiosis, and by day 1, 2, 3, we measure if we could find bacteria in blood. This has been shown in humans with intestinal disease, but as you can see, there was not statistically difference between the days, so we cannot say that this is a valid test for the moment. Now, it has been very well said yesterday and by Dr. Juan Carlos that we have a huge increase in the number of studies evaluating the microbiome, and that's because of the availability of new or not as new anymore technologies that allowed a lot of people to perform microbiome studies. So this is where I base my research. And before we start talking about markers, it's important to understand the whole of this, the role of this microbiome into the intestinal environment. So we know that we have a lot of bacteria, and horses have trillions of bacteria in their gut, and most of them are commensal, meaning beneficial bacteria. And I'm not going to repeat what has been said yesterday about the how important those bacteria are to digest fiber and produce volatile fatty acids that can be used as energy source. But one of the most important um, roles that those fatty acids play, it's also into stimulating a healthy intestinal layer that can produce many different molecules. For example, mucin, that is very important to maintain the mucus layer of the intestine. This layer is the one separating the most of the bacteria from the intestine and from the body. So if this microbiome of commensal are healthy and have a good composition, we are going to have a certain level of protection against pathogens. Um, and these bacteria are very important to keep those bad guys away, in a sense that they produce a lot of bacteriocins, but also they stimulate the production of volatile fatty acids and the tight junctions, and mainly they also stimulate the local immune system. Now, I'm not going to pretend I understand anything of immunology, but what we know is that if you change the composition of this microbiome, you also change the regulation of the local immune system, which is very important. Around 70% of the immune cells, they are located around the gut, which makes very evident the importance of this environment to maintain a healthy and working gut. So I like a lot this picture because he just showed us how the mucus layer is supposed to be. And so here we have the bacteria, here we have a nice mucus layer, and here we have the intestinal cells. But as soon as this layer is disrupted by different causes, then now we have this bacteria in direct contact with the intestinal cells, which obviously is going to cause certain degree of inflammation. Now, to understand how we can use those changes as markers, it's important to understand also the factors that can affect the microbiome. I'm just going to mention a few. But before that, I'm going to show you and get you familiar with the type of analysis we do for microbiome visualization. So this is what we call a PCOA, and basically represents the similarity of the microbiome. Okay, so if you have two dots that are close to each other, it means they have a similar bacterial profile, but if they are very separated from each other, they have different microbiome. So for example, here it's what happens when you give antibiotics to a group of horses. You have a complete shift of their microbiome, which some people can call that dysbiosis. Now, the age is one of the most important factors driving the microbiome composition. For example, here in red, you can see the composition of neonates, foals, and as soon as they start growing, we have a shift into this microbiome, and uh, it's important because depending on the age of the foal, you can have different markers, right? So here, as they start growing and becoming more similar to adults, we can see that there are microbial shifts. So if you give me a fecal sample from a horse, I can tell the age without looking at the animal, just by looking at the microbiome composition. So, Dr. Danesi and Calderon already told you that diet is a major driver of the microbiome. And based on those new studies, we can start to find markers that are associated with one type or the other of the diet. The other factors that can change the microbiome are, for example, the use of antimicrobials. So if we are developing new markers, it's very important to consider that if this animal, for example, had been treated with some drugs, but and also it's very important that each antimicrobial, it can cause a different type of shift or changes in the microbiome. So if one animal has been treated with TMS or ceftiofur or penicillin, you can expect that those markers will be different between those groups. Now, we know that horses are very sensitive. Everything you do can, stress them, and those can be associated with changes in the microbiome. So based on that, we started also to look into the disease and how the disease can change the intestinal microbiome. Uh, Again, Dr. Danesi started to talk to you yesterday about diversity, but I'm just gonna give you a little bit more examples how important and how this can be used as a marker for intestinal health. So the diversity, it's a little bit complicated because it's more or less a calculation. It's a mathematical formula that we use that takes into consideration the number of different species, but also how well they are distributed in their environment. For example, in those two communities here, we have the same number of species, I think 5 in each, but in community B, you can see that they are better distributed. Okay, so we think that this is important. And why we think this is important? For example, if you have a certain species or animal species that is feeding from one type of fruit in Amazon, and let's say that that tree gets a disease and they become extinct in the environment. So it's going to be very easy for those animals to start eating other types of fruits or nuts, so there's not going to be a problem for them. Now, if you are in an environment that the diversity is not very high, and some animals are drinking or eating from a certain species that becomes extinct, this is going to have a consequence. Those animals are going to starve, and the predators of those animals are also going to starve, and this is going to have a huge consequence in the environment. So for those reasons, we think that having a highly diverse microbiome, meaning a lot of different species that are well distributed, we think this is the best for the gut environment. And what we have been seeing is that the diversity is usually decreased in horses with several different types of diseases. Okay, so the problem in measuring the diversity is that to do that, you need to do microbiota sequencing analysis, which it's still expensive, takes a long time to get the results back, and we're gonna see that it has other types of limitations. Now, I have a particular interest into equine colitis, and as you can see, diarrhea also decreases the diversity of horses, so it can be used as a good marker for dysbiosis, but also it changes the composition of the microbiome. So horses with diarrhea, they have a different type of bacteria than healthy horses. And this has been shown repeatedly by other groups. Here, for example, from Caroline Arnold from Texas, where she shows that healthy horses have a different type, and especially if they have antibiotic-associated diarrhea, they have even more accentuated shifts and also lower diversity. Now, colic, it has been also shown that to decrease the diversity of the intestinal microbiome of horses. And also, we are starting, as new studies come up, we are starting to find what type of bacteria are more associated with healthy animals and which ones are associated with disease like colic and colitis. Now, this is a very nice study from Scott Weiss where they show uh, increases of Pseudomonas before those brood mares had become showing signs of colic. So they suggest that you can actually see changes in the microbiome even before they start with the clinical signs, which in this case we're going to discuss. It could be, for example, used as a marker to detect colic before it even happens. Now, I have been collaborating with different studies in colic. This one has just been published, and what we could demonstrate is that according to the type of bacteria, you can differentiate the type of colic if it's inflammatory origin or if it's small or large intestinal origin. And we also follow up those horses from admission until day 5 of hospitalization. And as we can see here, it's very clear that the dynamics of some of those populations change during hospitalization. So maybe in the future, if we can do a test that is quicker and cheaper, we can kind of monitor those populations that we know we should be low before you can send your horse home. So let's say we measure the amount of protobacteria, and now we say your horse is good to go home because they are into the reference levels. So those are only initial explorations of these possibilities, but maybe in the future we are going to be using those kind of tests in a clinical basis. Now, in this other study, I also collaborated from a group in Brazil. This is unpublished, but Julia Arantes, what she did was also to compare different types of origin of the colic, and that was in agreement with the group from Belgium that I just showed the results. And what she also shown is that according to the or place of the origin of the colic, you can have a different diversity, but also different composition. And what's very striking here is the difference between the evolution of the colic. So if you have more than 24 hours, you have one type, or the microbiome starts to shift a lot compared to acute colics that are less than 24 hours. And also, what she did, which is worth mentioning, is that she went back into the farm of those patients 6 months later to see how the microbiome has recovered, and it did, compared to a group of healthy horses. So, but when you look closer, you can see that there is still a statistical difference in the composition of horses 6 months later after they had surgical colic. So our hypothesis is that those animals, they persist with some degree of dysbiosis even 6 months later after the colic episode. And what we are going to do now in the next episodes is to see if those abnormalities that remain in the intestine, they can be related to the occurrence of recurrent colic. Now, let's talk a little bit about the limitations of microbiota analysis. Uh, the major problem is that we deal with relative abundances, meaning, for example, one exercise here that I can offer to you is what's the relative abundance of Pseudomonas in this environment? So you can see that's 30%. But if you look at this other environment, we can see that the relative abundance is the same, but you still have a lot more Pseudomonas there. And it goes on for all the other populations and bacteria. So we can have two different analyses that are giving us 30% of Pseudomonas, but one environment has 30,000 and the other one has only 3. So when we look into studies that report an increase of certain bacteria, we have to interpret that with caution because it might not be true. For example, in these two environments, I have the same number of Pseudomonas, but the abundance in the second sample it's higher. Okay, but we still have the same number of bacteria. So we have to interpret that with caution. The best way to analyze a sample is to use quantitative measurements, for example, quantitative PCR. Okay, and sorry, I just realized the title of this slide is in Portuguese, so well, I guess you can understand that. So, for example, this is real data for Enterococcus, and we can see that if you look at the percentages into comparing healthy horses and horses with colitis, we can see that there is a lot more percentage of Enterococcus in horses with colitis. So you might be thinking this is a good marker of colitis because it's higher in horses with colitis. But in reality, when you get the same samples and you quantify them and you look at absolute quantification, you're gonna see that in numbers, real numbers, there is more Enterococcus in healthy horses than in horses with colitis. Okay, so next generation sequencing is good to find um, which bacteria might be increased in each environment or in each situation or each disease. But now we are at the stage that we had enough studies using next generation sequencing, and we need to start validating this using quantitative analysis. Okay, one classic example is if you read studies looking into intestinal inflammation, you're going to see that they claim, including my own studies, we claim that lactobacillus it's a good marker for inflammation because the relative abundance it's elevated. But when you do real quantitative absolute quantification, then we see that there is no difference between healthy horses and horses with diarrhea. And that brings us to the several services that are now offered for microbiota analysis and detection of dysbiosis. Again, be mindful that those analyses might not be as easy to interpret. Like I get many requests to look into results, and many times I'm tied to say, I'm sorry, I cannot help you because this is all relative abundance. So be sure that the test you are choosing, they also have some degree of absolute quantification. Now, considering that my PhD student, Laurie Boucher, and again, this has been submitted for publication, but has not been reviewed yet. She has selected, and we are also working in collaboration with the University of Guelph and some other researchers from Texas, where we are comparing, or we have selected a group, a set of bacteria that we believe can be used as good markers to detect intestinal dysbiosis. So here I give you some of the examples that we found that were statistically different between healthy horses and horses with colitis. We are analyzing them separated in Canada and US, so left Canada, right US. And as you can see, we have been testing a whole lot of bacteria, and we have some promising results. For example, when we use this data to create a model or a PCOA based on the composition, only based on those statistically significant, here we have 8 species, we can see that we are able to cluster the samples from healthy horses completely or statistically separated from the horses with colitis. And we can also differentiate. For example, here we are using healthy controls from Quebec and from Ontario, and we can see that even if we only look at healthy horses, based on this test, we can already differentiate the origin of the sample. And the same is true for the samples from the US, where you can differentiate if the horse has antibiotic-associated diarrhea, diarrhea caused by salmonella, or if they are healthy. So this is a small sample set, but we are already able to have very clear and statistical differences. But as we start using and offering this kind of tests, we can in the future, for example, look into, we can explore, for example, the degree of severity. Let's say the horses that are outliers into our analysis, they might have a very clear separation in our PCOA, and this way we can, in the future, of course, we can tell the owner, your horse has a poor prognosis or it has more severe dysbiosis, and therefore we recommend this or that treatment. And again, like I just said, in the future, and we are starting to get more and more evidence that we can separate and find changes in the microbiome that can predict different outcomes. In this study from Clémence Loblier from in Belgium, she was also could find that horses that survived from infectious cause of colic, meaning diarrhea, they had different markers into the feces compared to the horses that survived. So again, this is all very preliminary, but as we start to get more data and start to validate that, maybe in the future we can look at the microbiome and say your horse has 80% chance of survival. And also what I find very important as a future perspective is that we look into this type of analysis to consider each horse as a different individual. So we have a panel now that we analyze 8 or 10 bacteria, or in the future we can include even more. But you know, you can create a model to tell how bad the dysbiosis is, but also, in my opinion, the most important is to look at the individual level. So one horse might have, um, um, you know, when you look at the beneficial bacteria, one horse might have not a lot of Lactobacillus, so or not a lot of Turicibacter. So this specific individual, it might benefit from a target therapy where we can design a kind of a probiotic containing only the strains of bacteria that this horse is deficient, and now we can treat them in a more precise manner. And also, the results of this research had led us to make further explorations. For example, we see that we know, we already knew based on culture that horses, they have, even healthy horses, they have a lot of E. coli in their feces, but um, so does so do horses with diarrhea. But when we look, we had this hypothesis that maybe the species of E. coli that are in healthy are different from the horses with diarrhea. We don't often or ever consider E. coli as a cause of diarrhea in horses. We don't test because if we grow them, uh, it means nothing. But with these results, what we saw is that we also try to test for the presence of virulence genes. And we just found that in healthy horses, the E. coli, they don't have a lot of virulence genes, but a subset of horses with diarrhea, that's where the majority of the virulence factors were. So with that, we are starting a new prospective study in where we're going to try to culture those strains, and we are going to do some further analysis to try to claim or to investigate if E. coli could actually be causing colitis in a certain group of horses. Now, again, we don't need a test to tell me that my horse has dysbiosis. If I can see diarrhea, it's obvious that it's going to have dysbiosis. But there are other uses of this type of analysis. For example, in this subset of horses that were treated with antibiotics, and this is the same for dogs and humans, there are always some individuals that we say that they have a low resilience of their microbiome, meaning that they're going to stay in dysbiosis for a longer time. Look at this horse here under the arrow. So he was treated, all of the other horses recovered the bacteria by day 21, but this guy, he stayed in a dysbiotic state. So if we can try to use this kind of analysis to detect and maybe in the future make associations with susceptibility to diseases, then we can start to consider that as a good marker for intestinal health. And like there is no limit to where we can use this kind of analysis. For example, this work from Nuria Mack, living in France, working in France, she has been also detected or making associations of good bacteria with good phenotypes of foals. So in the future, if we can test for the presence, we have to validate this first, but then we can test for the presence and quantification of this bacteria, and we might start treating or using microbiota manipulation to actually improve phenotype of those animals. And also detection of early disease in cows. You can find microbial shifts just before they start or they develop clinical signs of certain disease, like Sarah. And maybe, like Scott Weiss has shown, we can also prove that in horses and find which animals are more likely to develop disease. Now, take-home messages. We have been using traditional markers like lactate and calprotectin to measure intestinal integrity. So those are markers that are valid, but as we have discussed, they have some limitations. And we have been exploring oops, what did I do? Sorry. Can you still see my screen? Maybe change the zoom. Sorry, can anybody tell me if you can see my presentation? I'm almost done anyways.
Yes, Dr. Marcio, we can see your
Okay,
presentation.
perfect. Okay. So as we start to work on the development of those new markers, and we still need to validate them, and they need to become more accessible. For example, the new markers that we are testing now, they are very easy to perform a qPCR because it takes a few hours, plus a DNA extraction, and they don't cost a lot. So this is something that could be used in a clinical settings. They have been using that, especially our collaborators from Texas. They have been using that in dogs. They develop what they call a dysbiosis index, where they can get the sample and sometimes return the results on the same day to the clinic. So this doesn't cost a lot, but again, it needs to be validated. And it's very interesting that this could be used, for example, in horses with diseases that are not very obvious to present with dysbiosis. For example, horses with inflammatory bowel disease, or horses that are off feed because of some level of intestinal inflammation. And maybe in the future, we won't need to do a surgery to take a full thickness biopsy of the intestine. And if we can, based on changes in the microbiome, help to the diagnosis and also to establish prognosis of those diseases, then this can be very, very important to equine medicine, right? So, for future perspectives, we can consider the development of new strategies. For example, I also work with the development of new probiotics. And when we look at the shifts of the microbiome composition over age or over diet, we clearly see that the major bacteria in the gut are not the ones that we use as probiotics, commercially available probiotics. So, for example, if you find a lot of streptococcus in foals, what does that mean? Is it a normal finding, or is it good? Is it bad? We still don't know. But sometimes I find some people consult with me and they give me results of a test where they found a lot of Clostridium, and the diagnosis is inflammation caused by Clostridiosis. But the test it didn't allow the identification of species. So we not even know if those species are commensal Clostridia or they are pathogenic Clostridia. So we know that a big percentage of the microbiome in adult horses are composed by Clostridia species. So I have to disagree and say I cannot tell there is inflammation. I cannot tell there is a Clostridiosis going on. What I can tell is that maybe this microbiome of your horse is very healthy because it has a lot of Clostridium. So, and we never consider to use Clostridial species as a probiotic to correct this biosis. And my final remark is that we also are doing some studies where we are trying to identify the intestinal microbiome of horses at the species level. And what we are finding is that most of the species that are there are still unknown. All the analysis we do, it allows us to go to the genus level, but when we try to go further and see which species are there, we cannot. All of them become unclassified, meaning that there is still a lot of new species that we need to discover to improve the current data banks, but also that might be very important to treat those animals in the future, and why not to be used as a good marker. With that, I would like to thank you for your attention and for the opportunity you gave me to share some of my knowledge with you, and I am available for questions. Thank you very much.
Hello, Dr. Marcos. I can see you.
Oh, I can see you now. Good to see you.
Thank you very much for this impressive presentation, and thank you very much for sharing with us this successful scoop to NAEQUINA. I think this biomarker could help us also to owners and trainers to understand that any feeding management in horses, I mean, if you offer any kind of forage or any kind of concentrate, have a direct impact in the microbiome.
Absolutely. If you change the type of fiber used in the composition of your diet, you're gonna see changes in the microbiome. And it's very important you mentioned that because some
Dr. Marcio,
studies.
I have a question.
Yes.
Sorry, I have
No,
a question.
no.
How many months or years do you consider that this biomarker could be in the market?
Uh, well, it's ready. It's ready to be offered. We submitted for publication, and we are waiting for the comments from the reviewers. As soon as they say we agree, it's a valid, then we can start. But before offering in a commercial way, I would like to first validate because right now it's validated for horses with diarrhea, right? So is it useful to test or to use in horses that are not doing well, in horses that have been treated with antibiotics. We are actually also testing uh, how valid the test is to see recovery of the microbiome after the use of antibiotics. So, I would say in 1 year or two, we're gonna have enough data to. And again, if you want to do and to pay, I'm happy to offer you. But I'm always very clear to say it's very hard and still to interpret the results until we have a large data set that allow us to do that.
Okay. We're going to wait in this biomarker, Dr. Marcio. Dr. Marcio, an attendance sent me a WhatsApp message directly because she is working with microbiome, and she want to ask you that in the case of old horses, some of these horses have some inflammatory biomarkers because of their past excessive exercise or bad feeding.
Mhm.
Do you think that this biomarker could help these horses? They are
Yes.
held more that they
Yeah.
continue with having more years in this era?
Well, yeah. It's a very important question. So what we see with biomarkers is that we are able to detect some degree of inflammation in the gut. Okay, if you have inflammation, you change the composition of the microbiome. For example, especially when we talk about proportions. So, for example, E. coli or Enterobacteriaceae in general, they are good markers of inflammation. So, if you test an older horse and you see that it has a lot of E. coli, or the species of E. coli are not the ones that should be there, then at this point, this kind of analysis can help those horses. And as we evolve, we talk about different strategies. For example, there are some people working with phages. Phages are viruses that can be specific for some species of bacteria, including E. coli. So let's say you have high abundance of E. coli and you want to treat your horse, you give them some viruses that will attack those E. coli and will decrease the amount of E. coli. We talk about fecal microbiome transplantation, which I have doubts how well they work in horses still. But there are different strategies that we can do that could eventually help those horses.
Okay, thank you, Dr. Marcio. Continue with this question, Dr. Marcio. The same person asked, you you said that this biomarker can prevent some diseases in horses.
Mhm.
Um, how far in advance can these diseases could be prevented in horses?
Yeah, that's a good question. And again, when we read the paper in broodmares having volvulus, we have serious methodological limitations with that study. But so those are initial findings that you could further explore. Okay, it's very difficult to do a study like that because you have to collect samples before they get sick. So you have to collect samples from a huge number of animals and then
Mhm.
go retrospectively and analyze only the ones that got sick and the controls. So they did a very good job on that sense, but we still need a lot to validate that. What I can give you, Elias, it's an example of a study we are doing in cows. Okay, after the calving, we followed the uterine microbiome over time, and we saw that cows that developed metritis, they had some differences in the uterine microbiome compared to the cows that remain healthy and that became it's also associated with fertility. So right now we are testing our findings in a larger number of cows, 200 cows, where we are also using a kind of a test, a dysbiosis test, like the one I show you, but for the uterus of the cow. And I don't have results yet, but I hope because those samples are collected before they get sick. So 3 weeks after parturition, and now depending on those changes, I am hoping that I can tell you with some precision which cow will become sick with endometritis and which cow will remain healthy. So this is huge. This can be huge, right? If the results are good. So I hope I'll have, and in the future we can do the same things for horses. We can adapt our
Yeah.
markers according to the kind of disease. So if I'm treating them with antibiotics, we can incorporate new markers that can be affected by the use of this or that antibiotic, and according to the different kind of susceptibility. Let's say you're going to do a diet transition in your horse, and you can be sure that the good bacteria will adapt in the good way, so you can follow up the microbiome transition of during that diet change.
Okay, Dr. Marcio Costa, thank you very much. We have two messages in the chat, and we have a final question for you, Dr. Marcio. The first comment we have of Ana Maria, and she said, thank you very much for your presentation, Dr. Marcio. It is so important. This work is also important in humans and has been done for a long time. This is the first comment. And we have another comment of Dr. Silvia Bundings of, do you remember her?
Yes, yes.
She's also a coordinator of NAEQUINA, and she stayed also in Mexico.
Nice
with
to see
her.
you all here.
She said, Dr. Costa, it is a great to see you again and listen to your interesting talk. Do you know if there is a significant difference in the microbiota of horses grazing and horses receiving hay?
If it's if it's a significant difference in the microbiota of horses receiving hay, is that the question?
Yes. I mean, did you know if there is a significant difference in the directly in the microbiome between horses feeding grazing or that horses receive hay?
Oh
I mean,
yeah.
natural behavior
Yeah.
versus
Yes, there is a
hay
big
offer.
difference. Yeah, everything will change. The type of fiber, the ingestion of water, the hydration of the intestinal content, and also the availability of different the composition of the diet, like vitamins and nutrients in the hay compared to the pasture, and also varies according to the age of the pasture. You know, if it's more mature or young, you're gonna have a different composition of soluble carbohydrates. So all of those things can change, and this is very clear. For example, here in Canada, where we don't have pasture during the winter, right? They cannot graze on the snow, so they are 100% fed with hay, and we see an impact of the season in their microbiome. Yes, so the answer is definitely. Good question.
Thank you, Dr. Marcio. We have another question for you from Ariel Pellegrini. Ariel asks, in which type of samples do the biomarkers use?
Yeah, so the only one we have are fecal samples. Okay, that's the easiest. That's the only one we can get, unless you go to by endoscopy to collect duodenal sample. That could also be done, but nobody, as far as I know, has validated the composition of the duodenal microbiome related to diseases. So it would make a lot of sense if you have, for example, a horse with IBD or with a lymphoma that could, cause a lot of inflammation in the proximal intestine, and this could change the microbiome. And that's part of the study we started with the capsules, because we wanted first to validate if we could have a good sense of where the capsules are collecting samples in the intestinal tract. And then the next step would be to evaluate horses with IBD to see if the duodenal microbiome could be a good distinctive from compared to healthy horses. So, but right now we do the analysis from feces, which is a good proxy of the large column, I would say.
Mm.
It's quite different. If you
Okay.
go deep into the
Yeah.
composition, it's quite different, but that's the best we can do.
Okay, Dr. Marcio, thank you very much. Dr. Marcio Costa, you said that this biomarker is in the market. The same. I have another question. Where we can buy this biomarker, or do you have any information where we can
No,
have it?
you cannot buy, and it's not in the market.
Okay.
What I said,
Bueno.
we are developing in our lab in collaboration with
Okay.
Caroline Arnold from Texas, and it's not offered yet, okay? But we know how to do, we can do, and there are some. What people ask me all the time is that, can you do a test in my horse? So there are some stabilizers that you can buy, or you can use alcohol, for example, where you can put a little bit of feces, and then you can ship that refrigerated to my lab, and then we can analyze that. But I'm not advertising that because I don't have the capacity to run and to give you the
Yeah.
results, and also I still don't know if the results are meaningful or not. So I'm not responsible to offer a test that it's not meaningful and can actually um, improve your horse's life. So right now we are at the stage of validating everything before we can claim that it's a good test.
Perfect, Dr. Marcio Costa. It is a really pleasure for having you again here in NAEQUINA, and we hope we can have you again in Mexico, Dr. Marcio.
Anytime. It will be my pleasure.
Um, so we don't have any more questions, Dr. Marcio. Thank you very much. We really appreciate your participation, Dr. Marcio.
You are very welcome. So, what do you recommend that we go back to the main to the main session, or?
Yes, Dr. Marcio, please.